the new language of amino acids instead of proteins. And then we're going to talk about how these the proteins that are produced, how they are properly folded into those three-dimensional structures that are required for biological activity. And in some cases, towards the end of the chapter, how they're extruded outside of the cell. Okay. Perhaps this is what you need to write down, like the loaders, clamps, and DNA synthesis. So we're talking about an accessory protein that supports the RNA polymerase. And the accessory protein that helps the RNA polymerase dock onto its substrate are these sigma factors. And they're analogous in terms of kind of accessory proteins that we just talked about, these clamps and loaders that we talked about in Chapter 7. So we're going to be talking a fair amount about RNA polymerase. And when we use the term holoenzyme, it is not quite ready for prime time. It needs to be completed in some way. The example completion here is that sigma factor that helps it load onto its template. And like I said, so these sigma factors are required to bind that RNA polymerase onto what it is going to transcribe the DNA into RNA. Okay, here's a really nice figure. I think the only thing that you should write down is just what is this red thing. The sigma factor is shown in red. That is what is not on your outline. But just refer to figure 2 in this, and you'll see the marvelous RNA polymerase molecule. I'm not going to require you to know that it has all of these interesting subunits of separate polypeptides. So but understand, RNA polymerase is a nice example of quaternary structure where it requires multiple protein subunits to aggregate together to give it its function. And then just see the physics of this. It has this marvelous crevice into which perfectly fits a single strand of DNA. And it is this sigma factor that docks this protein onto the DNA template to transcribe it into RNA. So it's a really nice figure showing the three-dimensional relationship. And then basically what's shown here in A versus B is the front side where you can see the groove in the protein in which the DNA fits, and then simply the back side. So the groove is on the other side. So just two different angles of the RNA polymerase being shown. And then just to reemphasize, the sigma factor is what guides that RNA polymerase to the correct sequence, okay? Not just to any part of the DNA. Under certain conditions, it is that gene that needs to be transcribed. And so it's the sigma factors that say, under anaerobic conditions, we have to synthesize this enzyme to allow us to respire in the absence of oxygen, not this enzyme. So the selectivity of what genes are expressed and what genes are not expressed has to do with the sigma factors that bind the RNA polymerase. Okay? So it's the same RNA polymerase that's synthesizing aerobic genes and anaerobic genes. It's different sigma factors that lock them onto those two different genes. So really important as far as control of gene expression. Okay, so the promoter is this section upstream, and I think we showed a diagram of genetic structures, and I think you've seen them before. The promoter is upstream of those structural genes and is where that sigma factor docks that RNA polymerase onto, which signals the beginning of the gene. Housekeeping sigma factors. So this sigma factor is used in all sorts of operons of genes, and that's the use of this term. Housekeeping, it's just for a whole broad group of sequences. There are these consensus sequences when we use these terms, minus 10 and minus 35, that means minus 10 bases upstream from the beginning of the gene and 35 bases upstream from the beginning of the gene, because the gene starts at plus 1. And so when you see any negative numbers, you know it's upstream in the promoter control region, upstream of those structural genes. And then, like I've said, a single bacterial species can make several different sigma factors because each sigma factor is specific and will be turned on under certain conditions. Aerobic versus anaerobic is my example. So this is not detail that you need to know, but it is, cool information. Right here, plus 1 is the beginning of the gene. And it actually shows many genes kind of all lined up next to each other. So they're going to be completely different sequences here. But then in minus 10 base pairs and minus 35 upstream of the start codon, of the start of the gene, are these control regions. And so looking at then the consensus of all of these control genes, what do they have in common with each other, which is consensus? You can then look at the consensus sequences. They all start with TT, TG. And so then there is a section that can vary a little bit. And then they end up in AT. And this is the minus 35, and then the minus 10 is conserved with these genes. And I remember the first time I found, it's actually called the Tata box, because this minus 10 region is usually Tata, or Tata. And I remember I was looking for a particular gene, and I've done a ton of sequencing, and I couldn't find it, and one day I found the Tata box, which said, this is 10 bases away, all I have to do is move 10 bases, and here is my gene for nitrite reductase. And it made my day, and it made my postdoc, and it got me this job here. Because we published that paper, and all things went better after I found the Tata box. Anyway, which are conserved sequences, which is where these sigma factors and RNA polymerase bind to then synthesize these genes here. So all of this is on your outline. There's no, I didn't add anything here. And then maybe I'll close with this. I still got about 5 minutes. Everything is on your outline on this slide, and it's how sigma factors recognize specific DNA sequences. These illustrations are getting better and better. So remember, the orientation is, here is your RNA polymerase, and here is the beginning of the gene, and the gene that you want to transcribe is in light gray, and then the other strand you're not transcribing under these conditions. But here's the beginning of the gene. So upstream at the minus 10 and minus 30 section, or minus 35, you have this really interesting interaction between the sigma factor, which is in red, recognizing that particular sequence in order to transcribe those particular genes, and helping the RNA polymerase dock onto its template. So these accessory docking proteins, sigma factor in this chapter, clamp loaders in the previous chapter, are really important to help orient these otherwise big, sort of clumsy RNA polymerase molecules, which are like, lead me to my work today. So without these sigma factors to dock them, and it's a gorgeous, fairly realistic diagram. Here's your double-stranded DNA and the globular shapes of these proteins, and then this massive magnesium-based RNA polymerase. Just really, really nice. So upstream, downstream, kind of orient yourself. These are your genes that you are synthesizing. Okay, so this is the big picture view, and this is about all I'm doing with transcription and translation. We've had this before. So with the help of the sigma factor, initiation of that transcript is carried out by RNA polymerase. And once it binds to its substrate, it is the real enzyme, but it's the promoter region to which it binds. Then it starts transcribing DNA into RNA. And so you get a longer and longer chain of RNA that is extended, and then you reach a stop codon at the end of the gene, which then detaches the RNA polymerase from the DNA after the transcript is made. So, understand that this is the big picture view. We're transcribing DNA into RNA. You've learned that before, but that I just give you the outlines for you to make that connection. Okay, we'll talk about this on